So now that we are done with all the musculocutaneous disorders and all the anatomy, etc. So now let us move on to the physiology part of the human body. Okay. So to begin with, we'll discuss the general physiology alone here. The rest of the physiologies for that particular systems we'll be discussing in systemic discussions. Okay. So now let us focus only on the general physiology. So there are only three main topics that we have to discuss here in general physiology. One is the types of transport, the other one is the cells, the fluids, and the nerve physiology. Okay. So let us start the topic from transport. Okay. So there are three kinds of transport. One is active transport, one is passive transport, and another one is facilitated transport. Active transport is of two types. One is primary active transport, the other one is secondary active transport. Primary active transport is where the channel itself utilizes a ATP. Okay. So we know the famous example of sodium potassium ATPase pump, right? So this is a pump. Okay. So there is a material that has to be transported from outside to inside, okay, or from inside to outside. Okay. So what happens is this transport itself utilizes a ATP, meaning ATP is nothing but a currency. Okay. So currency is 10 rupees. Okay. So it gives 10 rupees or utilizes 10 rupees. So, and then transports this particular material into inside or probably outside. Okay, it doesn't matter. Okay, basically what action it is doing is nothing but transport of that particular material. Okay, so it is utilizing the transport by itself, whereas the secondary transport is one of the pump is utilizing the ATP. Using that ATP, the other pump the other substance which is not in that particular pump is also getting transported. Okay, utilizing the ATP from or utilizing the pump mechanism of other pump, this other substance is also getting transported. Example is sodium potassium and chloride pump, or sodium calcium pump, or sodium glucose co-transporter. Okay, so here the sodium glucose co-transporter is SGLT1 and SGLT2, where the sodium is getting transported using ATP, but the glucose is also going along with it. So because of the pump mechanism of one transport, the other substance is also getting transported. So that is the basic mechanism with respect to this primary and secondary transport. So this is primary and secondary active transport. You got it right. So basically active transport meaning there is utilization of energy. So examples are what is important here. Okay. Then moving on to passive transport, it is diffusion of salts or any other water, everything will be through diffusion. Okay, this is a passive method of transport. Then there is one more method where there is facilitated diffusion, where using a transport protein the substance is getting transferred. Suppose this is glucose and it is getting transferred into the other part. Okay, so from above the cell membrane or from the other part of the cell membrane, it is getting inside. Okay, from outside to inside, or let us take inside to outside. Okay, so best example is GLUT transport. Okay, so GLUT transport is what has been the focus of examiners. Okay, so because different types of GLUT transport are transporting in different kinds of tissues. Okay, so let us discuss one by one. GLUT transport 1, GLUT 1 will be the normal glucose uptake. Okay, so normal glucose uptake will be done by GLUT1. This is mainly, so remember this example please, brain. Okay, mainly brain is using GLUT1. Okay, brain is using GLUT1. Please remember that. Then the rapid uptake of glucose is done by GLUT2. Okay, GLUT2 is doing the function of rapid uptake of glucose, which can be seen in liver and pancreas. Okay, then coming to GLUT3, it is again same like GLUT1 is normal. Okay, GLUT4 is should be your main focus, because skeletal and adipose tissue intake is the main, that is where the majority of glucose should be required, right? That is where the majority of the action and all will take place. So that is why this is where the GLUT4 is. GLUT4 should be marked with star. Okay, so GLUT4 action. When asked which GLUT mechanism is skeletal muscle and adipose tissue is taking place, so it is GLUT4. Then GLUT5 will be fructose. Okay, so GLUT5 will be fructose. GLUT1 to 4 will be glucose. Okay, so this is with regards to facilitated diffusion. Okay, then I was discussing about the SGLT, right? The secondary active transport, where sodium glucose co-transport will occur. So here in enterocytes in the small intestine, it is SGLT1. Okay, SGLT1 is in enterocyte, whereas SGLT2 is in PCT of kidney. Okay, in kidney, you know that there is glucose reabsorption, right? 100% of the glucose will be reabsorbed. If 100% of the glucose is not getting reabsorbed, then you have diabetes mellitus. Okay, that is the main thing. So SGLT2 is in PCT of kidney, whereas SGLT1 is in the enterocytes. Okay, that's it with respect to transport. Then we'll see some cell physiologies. Okay, so in cell physiology, the main thing is the cell membrane. Okay, you should know about cell membrane and its physiology. Okay, in cell membrane, there is carbohydrates, there is lipids, and there is proteins. Proteins are the peripheral proteins, the integral proteins, and the lipid anchor proteins. Okay, so here the peripheral proteins, which are the proteins which give the structure to the cell membrane. Okay, so for example, a cell is there, its structure is maintained, the membrane structure is maintained by spectrin, the peripheral proteins. Okay, there are other structures called cytoskeletons, which we'll be discussing next. So those give the skeleton of the cell. Okay, but the cell membrane structure is maintained by the peripheral proteins. Okay, that's it. Then so one of the example of this peripheral protein is spectrin. Where does this spectrin come in? So spectrin can be seen when we are discussing about the RBC pathologies. Okay, RBC pathologies, this spectrin and all will come. For now, you please remember there is there are peripheral proteins. When we are discussing it in hematology, I'll tell you about I'll remind you that we had discussed in physiology about this. Okay, so there is spectrin, dystrophin, which is a muscular disorder. So all these things will be Duchenne muscular dystrophy. So this is where the peripheral proteins will come in. Okay, that's it. Then there is integral protein and also the lipid anchor proteins. Then I was telling you, right, cytoskeleton is what maintains the skeletal structure of the cell. Okay, skeletal structure of the body, human body was maintained by the skeleton, right? Skeleton, bones and all we discussed in orthopedics, right? So that is what maintains the skeletal structure of the body, whereas the skeletal structure of one particular cell is maintained by the cytoskeleton. Okay, there are three types of cytoskeletons. Okay, microtubules, microfilaments, and intermediate filaments. So let us see what all these cytoskeletons do. What particular function this cytoskeleton do? Okay, microtubules has the function of cell division. Okay, so during cell division, you know that, right? These things and all that will come up during cell division. The chromosome are here, and so all this division of the chromosome, from 2n to n, all these things will be done by microtubules. Okay, these microtubules are the main target when we give some chemotherapy, because when there is no when there is no microtubule, then the cell can't divide and the cancer cells will die, right? Cancer is nothing but increased cell division. When there is no microtubule, the cell division cannot happen, right? So that is the main target. So the main chemotherapy drugs are vincristine, colchicine, all these things will act on microtubules. Okay, remember that. Then there is microfilaments, which we will be discussing further when it comes down to muscular physiology. So you know that, right? You would have studied in your first year and in your final years also actin, myosin, which are the muscle proteins. Okay, these are microfilaments. Then coming to intermediate filaments, these are the filaments that we use as tumor markers. Okay, certain tumor markers that we see whether the cell is dividing quickly. For example, if the cell is dividing quickly, there will be increased amount of intermediate filament, right? So that is when it acts as a tumor marker. Okay, so when the tumor marker is high, so that means that the division is very strong. Okay, so one more thing I want you guys to remember is microtubule and microfilament are dynamic. Okay, because they have a dynamic action, they divide and all these things, they'll change their places and all. So that is why it is called dynamic, whereas intermediate filament is static, it stays in one particular place. So that is the main thing that I want you guys to remember, dynamic and static. Okay, this is with respect to cell membrane and cytoskeleton. Okay, then with respect to cell junctions, there are communication. The cell junction is nothing but the space of communication with the next cell. So each cell to cell, the cell and cell communicates with each other. So there are types of cell junctions which maintain this communication. Okay, there are two types of cell junction. One is tight junction and the other one is gap junction. So usually most of the cell will be having tight junction, where there is no direct communication with the cells. Okay, so there is eccrine or merocrine, apocrine, autocrine, these kind of cell communication will be there, whereas the direct communication can be seen only in cardiac cells. This is very important. Please note that in cardiac cell there is you know that, right? In histology you would have learnt in first year and all, here there is intercalated discs and all. This is the gap junction, because in cardiac cell each cell to cell communication is very must. Okay, because for rapid action and rapid transfer of the electrolytes, there must be cardiac gap cells. Okay, so that gap cell is maintained by intercalated cells. So that is the cell junction. There are two types, tight junction and the gap junction. Gap junction are seen in cardiac cells. Okay, that's it. We are done with cell membrane, cytoskeleton we discussed, cell junctions we discussed, and then we'll go on to receptors and messengers in our body. This is a very important topic. Please note down this in your sticky note. Okay, receptors and messengers. Okay, so messengers to a tissue or a cell, what are the messages it might get? These are proteins, steroids and amino acids. Messages to the tissue or a cell will be in three forms. That is, direct protein can come, or a steroid or an amino acid can come and give a message to that particular cell. Okay, so these are the messengers. What are the receptors? So in a cell, a message has come. Okay, to receive that message, the cell has cell surface receptor, intracellular receptor or a nuclear receptor. Okay, so let us divide that broadly as extracellular or the cell surface receptor and intracellular receptor. Okay, so let us discuss about intracellular receptor. So intracellular can be cytoplasm and nucleus, right? So if the in cytoplasm, the adrenal hormones will come and act. Okay, and if it is thyroid hormones, it is gonna be nucleus, that is the receptor. Okay, so for example, TSH has come in, or so all these TSH the stimulating hormone for thyroid will come in, right? So those hormones or those proteins, so it is basically an amino acid, right? Tyrosine or levothyroxine, all these things will go and bind to nucleus of a cell. Okay, and increase its metabolic activity. So what is the action of thyroid? We'll discuss in endocrinology, but for now the main function is increase its metabolism, right? Increase the cell's metabolism. So thyroid hormone will go and bind to nucleus of a cell. Then coming to adrenal hormones mainly go and act on the cytoplasm of a cell. Okay, all the cells in our body, its particular action will be there. So if it is the adrenal hormone, it is going to be the cytoplasm, it will go and act. Okay, so these are the two types of intracellular receptors. Then coming to cell surface, there are mainly three. Okay, there are mainly three types of receptors. One is G protein coupled receptor, JAK-STAT receptor, then MAP receptor. Okay, G protein coupled receptor is all the releasing hormones, like for example, let us take thyroid releasing hormone, all these things, right? Corticotropin releasing hormone. All these releasing hormones will go and bind to the cell surface through G protein coupled receptors. Okay, G protein coupled receptors. You would have heard different GPCR, G protein coupled receptors. It is nothing but this. In many questions and all, you would have seen this and you would have confused what the hell is this, right? So this is that. Okay, this is nothing but a receptor that is present on the cell surface of all the millions, trillions of cells that is in the body. These receptors will be there. Okay, when the releasing hormone is released, it will go and bind to that particular organ. For example, releasing hormone, so it will be definitely be there in pituitary has highest amount of releasing hormone receptor, because that is where all the hormones will get released from, right? So from hypothalamus the releasing hormone message will come, so pituitary starts releasing it. We'll discuss more about this in endocrinology, but for now this is the thing. Then JAK-STAT is growth hormone and prolactin. JAK-STAT receptor on the cell surface is where the growth hormone and prolactin will go and act. Okay, MAP is nothing but insulin like growth factor, like growth factor like. Okay, whatever the growth factor like is there will go and act on the MAP receptors. Okay, that's it. This is very, very important. Please note this table in your sticky note. So then there are different types of secretions, okay, from a cell. Okay, the types of secretions will be discussed here. So there is merocrine type of secretion, apocrine type of secretion, and holocrine type of secretion. What are these? Merocrine is nothing but a cell is there and it wants to send a signal to the next cell, or it wants to release something. What it does is it releases the vesicles. Okay, if it releases a vesicle, then it is known as merocrine. Okay, merocrine type of secretion. If the cell apical portion itself gets detached from the cell, then it is gonna be apocrine. Apocrine, okay, is the type of detection. Suppose if the whole cell is getting broken down and forming a messenger, then it is going to be holocrine, whole cell. If it is apical, apocrine. Then if it is the vesicles that is secreting, then it is going to be merocrine. Okay, what are the examples? Merocrine is going to be salivary gland. Apocrine, the apical part, will be the mammary gland, whereas whole cell is sebaceous gland. Okay, that's it. This is all and everything you have to remember with respect to the cells. Okay, that's it. Then we'll move on further to the fluids. Okay, fluids, these are all fact based questions. Okay, so fact is total body water is 60% of the body weight, wherein which 40% will be intracellular fluid and the 20% will be extracellular fluid, and the 40% cellular structure remaining. So 60%, 40%, 60% will be ICF and ECF, whereas the rest 40% will be cellular structures. Okay, this is a fact based question. Then coming to DY diagrams. So what are these DY diagrams? So it basically tells what the fluid level or the fluid concentration with respect to the normal concentration or the normal fluid level is. Okay, so I told you, right, in the total body water it is gonna be 40% ICF and 20% is ECF. So this is the normal thing or the normal box, let us consider. Okay, ICF and ECF. Okay, suppose if there is isotonic fluid loss. So now the diagram differentiation is based on whether there is fluid loss or fluid gain, whether the concentration is increasing or whether the concentration is decreasing. Okay, so suppose if there is isotonic loss, there won't be any concentration change. Okay, so the y-axis, this is not a graph. Okay, so the y-axis gives the concentration, whereas the x-axis, okay, so this is not a graph, don't think it as a graphical representation, just this is in a x and y, so I'm just telling the axis that you have to be aware of. Okay, so y-axis is concentration, whereas the x-axis will be the volume that we are discussing about. Okay, that's it. So now that you got the orientation of this diagram, so let us see in different case scenarios what this diagram will happen. Okay, so if there is isotonic loss, then there won't be any concentration difference. Okay, this is an isotonic fluid. Okay, so there won't be any concentration difference, so the y-axis will be normal. Okay, y-axis, there won't be any change, whereas in the x-axis there will be the loss of fluid, right? So loss of fluid will always be from ECF. Okay, loss of fluid will always be from ECF, so that is why the volume has decreased. Okay, from 20%, this box represents a fluid level. Okay, so the fluid level in this box has been reduced, is the what is the graph representation that I have given here. Okay, so I'll discuss the examples later. So let us now discuss what are the graphs first. Okay, then if there is a isotonic fluid gain, then the ECF volume will increase, right? So from 20% it has become more when there is isotonic volume, so there is no change in concentration. So remember, so that is why when there is a gain of isotonic fluid, then the ECF will increase. Okay, that's it. Then hypertonic. So hypertonic loss. Okay, now hypertonic meaning the concentration, right? It is concentrated. So when there is a loss, there will be reduction in concentration. So that is why here there is a reduction. See, this was the normal level, but now the reduction of concentration is seen. Whereas with respect to volume, there is a loss of volume, right? So that is why the ECF has reduced volume. That's it. Okay, so this is the graph when there is loss of hypertonic solution, whereas gain of hypertonic solution, the concentration has increased as well as the concentration of the fluid level also has been increased. Okay, that is hypertonic gain. When it comes to hypotonic loss, so there is concentration has increased, where it has increased in ICF. Okay, please remember the concentration has increased in ICF. Okay, whereas the volume loss has occurred in ICF, because see, so what happens, hypotonic solution is being loosened out from extracellular fluid. Okay, so from extracellular fluid, the volume is getting getting leaked out. Okay, so what happens is from ICF, the fluid will start shifting towards ECF, because the salt concentration is more here. You know the osmosis, right? Osmosis meaning wherever the salt is there, the water will follow there. Okay, now that the fluid has been lost from the ECF, the volume from the ICF will move towards the ECF, because here more salt is there compared to ICF. So that is why the fluid volume will get increased with respect to ECF. Okay, then coming to hypotonic loss, hypotonic gain, see, the concentration is decreased. Why? Because it is an hypotonic solution, not a hypertonic solution. The concentration is decreased compared to the normalcy and gain. So that is why the fluid level also will be increased. Fluid volume is also increased, whereas the concentration is decreased. This is the dy diagram basic representation. Okay, so you got the representation, right? So now let us discuss what are the examples of these representations. Okay, so if there is isotonic loss, it is going to be vomiting or diarrhea where we lose isotonic fluids. Okay, and in gain it is going to be ringer lactate or normal saline is what is gain of isotonic. Okay, isotonic will be these things. Hypertonic solution loss meaning additions. Okay, additions where aldosterone is low. I'll discuss all these things in endocrinology. Additions, there is no aldosterone, so there is no sodium concentration. Sodium is getting leaked out, so that is why hypertonic solution is getting leaked out. Hypertonic loss is happening. Whereas when it comes to saline infusion, it is the hypertonic gain that is happening, right? So saline infusion is hypertonic gain. Okay, then coming to hypotonic loss, it is sweating or diabetes insipidus. Diabetes insipidus is nothing but losing the fluid, right? We are losing fluids from our urine, so it is going to be hypotonic urine that is being lost. Whereas hypotonic gain is SIADH, where there is no antidiuretic hormone, so there is no urination, so all the fluids start staying inside the blood itself or the body. So that is when there is increased hypotonic solution, because concentration has not changed but the fluid level has increased and the concentration is getting decreased. So that is how it becomes hypotonic solution. Okay, that's it with respect to dy diagram. Please note the dy diagram, all the solution examples. Okay, all the solution examples should go to your sticky notes. Okay, that is all with respect to dy diagrams. So now let us go on with nerve physiology. Okay, so the types of nerves are unipolar, bipolar and multipolar. In bipolar, the main example is retina. Okay, that's it. So moving on, there are types of nerve fibers. These are types of nerves. Okay, so now I am discussing about types of nerve fibers. Okay, there are three main types of nerve fibers: type A, type B and type C. Type A has four major subdivisions, that is alpha motor neuron, beta neurons, gamma and delta. Okay, so there are different functions of these nerve fibers. Okay, so let us see one by one. So alpha fibers will cause the motor function, whereas the beta fibers will cause the touch and pressure sensation. The gamma fibers will cause the reverse pain, where the delta will cause the pain. Okay, so the the pain sensation is being carried out by A delta fibers, whereas for example there is a pain in our body and it is carried to the brain or the spinal cord by A delta fibers. Whereas if I do a massage in and around the pain, so there will be a reverse of this pain, right? So that fiber is being carried by A gamma fiber. Please remember gamma fibers. Please remember this. Okay, A gamma is reversing the pain, whereas A delta is carrying the pain fibers. Okay, then coming to coming to B fibers, it is going to be the autonomic nervous system. Autonomic nervous fibers will be the B and C fibers. Okay, so autonomic fibers will be the B and C fibers that it's going to carry. So preganglionic will be B fibers, whereas the postganglionic would be the C fibers. What is this preganglionic, postganglionic? I'll be teaching you later. Okay, so autonomic system will be sympathetic and parasympathetic, right? So parasympathetic and sympathetic, when it comes out from the spinal cord, it is gonna be preganglionic. There will be a ganglion before it gets delivered as postganglionic fiber. Okay, so preganglionic and postganglion, both of these autonomic fibers will be in this way. Okay, so the preganglionic will be the B fibers, whereas the postganglionic will be the C fibers. Okay, that's it. So remember in this way. Okay, motor and sensory will be A fibers, whereas autonomic function will be the B and C fibers. Then the main and most important thing, and all the previous year questions are also around this thing only, the susceptibility of these fibers. Okay, susceptibility to pressure, hypoxia and anesthesia. Okay, so for pressure it is going to be A greater than B greater than C, meaning A will be more susceptible to pressure than B than C fibers, whereas in hypoxia B will be greater than A greater than C. Susceptibility of B fiber will be greater than A in that way. Okay, in anesthesia C fibers are more affected than B fibers than A fibers. So this is the main thing with regards to the nerve fibers. Okay, nerve fibers and what are its types and what is its clinical physiology or clinical significance. Okay, then coming to action potential of the nerve fibers. So we were discussing what are the types of nerves, types of nerve fibers and its clinical significance. Let us see how these nerve fibers actually carry a potential that is generated from the brain. For example, now I'm thinking of speaking, right? So I'm speaking. So how will my brain generate an action potential and it sends to the tongue or the tongue muscles. So let us see. Okay, so action potential, before going into how the working of action potential is, so let us see how it is measured. It is measured by microelectrode. Okay, so this is a previous year question, they have asked it. So measured by microelectrode. Okay, so then the main ion to generate action potential is sodium. Remember this. Okay, so sodium is the main ion that is helping in generation of the action potential, whereas the RMP, the resting membrane potential, is maintained by potassium. So this is the two electrolytes that are very important in generation and the maintenance of resting membrane potential. Okay, then now let us see how the action potential gets generated. Suppose I'm thinking of speaking, right? So the moment I think about speaking, my premotor cortex, the motor cortex, what precentral gyrus, the motor cortex of my brain, what it does is sends a so what it happens is sodium influx inside the neuron cells will happen. Okay, sodium ion that is present around it will start going inward. Okay, so this is a neuron, think of this as a neuron. So sodium ion starts going inside. This is the influx of the sodium that is happening. So then further moving down, the calcium influx will happen. What happens is once the sodium comes inside the cell, so calcium also starts coming inside the cell. Okay, without sodium coming inside the cell, the calcium influx won't happen. Okay, that is the thing. Then once the calcium comes inside, this is where the action potential generates. Okay, so it starts producing, and after a cascade of reaction, the snare proteins and all these things. Okay, so that is not much of an importance here. So after this cascade of reaction, the acetylcholine that was present at the end of the cell body, so or the cell junction, will get released. Okay, so after the calcium influx, the acetylcholine is getting released. Once the acetylcholine is getting released, the next neuron that was there with the receptors, so the sympathetic receptors or the receptors that is present after the first neuron will be having the acetylcholine receptors. Okay, this is called post synapse. Okay, this is the pre-synapse, this is the post synapse. Okay, so this post synapse will get the acetylcholine and then the cascade of reaction will start happening. Okay, so then we'll meet a final end plate potential or the final muscle. This is called end plate, where the final message will get delivered and the action of that particular thing will start happening. So this is how the cascade begins and ends up in generating an action. Okay, so the action potential waves that are generated now, so this will be different when it comes to the nerves and the nodal tissue and in ventricles. Okay, so the action potential that is generated will be different in these particular three tissues. So the action potential that I told you was from a brain signal to a muscle. Okay, so it will be very different when it comes to nodal potential, and it will be different from ventricle potential. Nodal and ventricle I'll discuss when it comes to cardiology. Okay, when we go to cardiology I'll discuss about it. Okay, now the major clinical significance I want you to focus on is the calcium influx. If I told you, right, all the calcium influx will happen after the sodium influx is happening. If this is affected, or if there is antibodies against this calcium influx or calcium channel, then there is a pathology called Lambert-Eaton syndrome. Okay, if there is an affected release of acetylcholine, it is because of botulinum toxin. Okay, more about this I'll talk about in microbiology. For now, you remember where exactly this botulinum toxin is acting on. Okay, if there is inhibition of acetylcholine release, it is because of botulinum toxin. If there is antibodies against the receptors of the post synaptic receptors of the acetylcholine, then it is going to be myasthenia gravis. Okay, you got the difference between Lambert-Eaton and myasthenia gravis, right? Okay, I hope you have understood finally. Okay, that is the major difference. Okay, that's it. Then I was telling about these nerve endings and receptors in dermatology, right? Where exactly these receptors are present. So this is what I was talking about: Meissner's plexus, Merkel's, Ruffini, and Pacini corpuscles. Okay. So what happens is, so when there is a fine touch, it is the Meissner's plexus that is getting the reception from, and if it is crude touch, it is gonna be the Merkel's. Okay. Remember, it is MR P, maximum retail price, right? M are two types. Okay, fine touch and crude touch, whereas the Ruffini will be the stretching. Okay, if I stretch a skin, it is going to be the Ruffini plexus that is carrying it, and if vibration is there, it is going to be the Pacinian plexus, that is Pacinian corpuscle, which will be carrying the vibration. So Pacinian corpuscle just looks like an onion. Okay, so remember this: Pacinian corpuscle just looks like an onion. Okay, this was asked in the previous year. Okay, if you want, just type in Google and you'll get an image of Pacinian corpuscle. So that is what I want you to remember. Then coming to muscle physiology, they have not asked much questions about muscle physiology. Rather than they have asked you to identify the sarcomere bands and what is the nomenclature with respect to muscle sarcomere. Okay, I want you to remember is MHAI Zara. Okay, my Zara. You know Mahendra Singh Dhoni, right? His daughter's name is Zara. Okay, remember in that way. My Zara. M is the M band, then H zone, then the A zone, I zone, and the Z band or the Z line. Okay, remember in that way, and you would be able to solve anything with respect to this. Okay, then other than that, the only two things that I wanted to remember with respect to muscle and neuromuscular physiology is the muscle spindles and the Golgi tendons. What are these two things? Let us discuss about. So the muscle spindles are which are present in the muscles. Okay, muscle tissues or the tendons. Okay, what happens is when the muscle stretches too much, the stretching muscle starts contracting. Okay, so and the antagonist to the muscle gets relaxed. For example, I am stretching my biceps too much. When does this stretching happen? So when I do a ankle reflex or biceps tendon reflex, so there is increased stretching of the muscle. Okay, not contraction, there is increased stretching. Okay, so when the stretching happens, the antagonist muscle tries to relax. When I'm like, for example, I'm tapping on the tendon of biceps, the antagonist muscle, that is triceps, tries to relax, and the jerk happens. Okay, so jerk in the opposite direction of the antagonist muscle will happen. So this is happening through muscle spindles, whereas the Golgi tendon is opposite to that, where when muscle is contracting too much, the contracting muscle will relax. So for example, I am so what happens is there, for example, I was telling you, right? So biceps, so I am contracting too much. I am lifting a heavy weight. What happens is, to protect my tendon, what it does is it suddenly relaxes it. Okay, so relaxes the same muscle. I am contracting the biceps too much, and the biceps suddenly relaxes without our knowledge itself. Okay, this is called Golgi tendon reflex. Muscle spindle is where too much stretch is causing the antagonist muscle to relax, whereas the Golgi tendon reflex is too much contraction is causing the agonist muscle itself to relax. Okay, that is the two things that I want you guys to remember. That's it with respect to general physiology. Thank you.